Thank you, everyone, for coming to my dissertation defense. It's really great to see all these faces and do a little bit of an unconventional presentation. So my dissertation is entitled Conservation and Comparison of Protein Interactions Across Evolution. And so first, I'll start with some bookkeeping. The agenda for the day is 3 to 4 is the public talk. If you have any questions throughout the talk, ask them to the group chat to everyone, not to me privately, and I will try and get to them. There's also going to be some time at the end left for questions. So if you want, you can wait till then. Or if you're confused on any point, just message the chat. 4 to about 5, I will be off in a different Zoom with the committee. But at about 5, whenever that's done, there will be a social at this same Zoom link. So if you want, you can just leave this Zoom running and then come back to it once you start hearing voices. Also, I would love people to keep their cameras on, just so I can see people and it's not talking to an empty computer. If bandwidth become an issue, we will adjust. Okay. I also want to thank my committee for all of their support over the years, particularly my PI, Edward Marcotte. You couldn't have a better PI or a better place to do your graduate work. It's been amazing. We also got Karen and Lulu and John and Klaus, who have all been really helpful and fantastic. So first, I'm going to start off with a quick overview of the papers and projects that I've worked on at UT. My first set of projects were working with a former postdoc, Benjamin Liebestein, at the lab, looking at some of the comparative approaches that have been used to study human disease genetics. So this is pretty much getting insight into human disease from multiple different organisms. And also work that we did characterizing some of the algorithms that tell which genes are corresponding across different species and characterizing some of their errors and strengths. I also worked on an orthology, which is these corresponding genes of genes in bacteria and yeast that were mutually replaceable, in that the bacterial version of the gene could cover the function of the yeast version of the gene. It was a fun project. My main work has been on the high throughput discovery of protein assemblies / protein complexes. I'll use those words, complexes and assembly, pretty synonymously. Getting started and learning from Kevin in his work building a map of human protein complexes, but then going off with Ophelia to do this pan-plant protein complex map, which is my main work and what I'm going to be talking about through most of this presentation. I also did some work with Klaus Wilke, who's on my committee, that started off during my rotation with Klaus when we noticed that a particular artifact of sample preparation was actually confounding certain evolutionary analysis of influenza evolution. And so that's been exciting, and that was followed up by Corey Dupai last year. And just the last little side project I worked on in graduate school was also with Klaus, working on accessibility tools and simulation tools of colorblindness for making scientific figures in R, and that was incorporated into a larger package for working with colors in R, just a programming language. Okay, so to start off, let's consider the following organisms. These are all eukaryotes, and they have extraordinarily diverse morphologies, lifestyles, number of cells from single cells to multicellular. And so we can all agree that it's easy to tell what's different about these things. You can kind of list them off, infinite numbers. But another angle to look at all of these eukaryotes, all of these related organisms, is to ask what is the same, essentially what is conserved and so basic to the functioning of eukaryotic organism that you're going to see it no matter where you look. So framing this as the core conserved biology. Multiple biological systems tend to be conserved across organisms. This includes processes like DNA replication that every type of organism needs to reproduce, pathways such as central metabolism, the universal need to convert sugar into energy so that we can survive, and finally, stable protein assemblies, which are stable interactions within proteins, sets of specific proteins that join up into larger three-dimensional shapes. So I'm going to consider these through the talk as our deeply conserved systems, processes, pathways, and protein assemblies. And so these are the types of biology that we're going to be finding across large swaths of organisms, and considering not the whole shape of the organism, but the molecules that make up the core function. No matter what your morphology, no matter how many cells you have, this is what's inside of your cell when you zoom in. So the function of is critical to the organism, but we still don't have a complete description of biology, how all the parts, DNA, your RNA, your proteins, your metabolites, your lipids, all of those things completely work together. As in, we can't simulate an entire organism and all the things that are going on inside. We don't yet know the rules of life, you could say. It's kind of like the rules of physics. What are the rules of life? However, one approach to learning these types of concepts is to search for common patterns in data from multiple organisms. So essentially, we're getting a lot of noisy inputs, combining and looking what you see, no matter what type of thing you're looking at. I thought of a metaphor of like placing 10 microphones in a crowd and trying to reconstruct like a difficult to hear loudspeaker announcement that maybe you couldn't make out individually, but looking at the overlap of all these noisy symbols, it can become clear. And it's the same thing with biology, your typical quality of measurement. So thankfully, species share thousands of genes with each other, which is good news for studying across biology. And the genes in humans that you're going to find across all these eukaryotes are, again, these basics. And the genes make a protein, and these proteins are the building blocks and the actors that are all working together in the system in order to build up the cell and keep it running. The conserved systems link phenotype, what's in our DNA, with our phenotype, or what we look like, or our morphology. So we start with DNA. DNA is transcribed into RNA, which is then translated into proteins. And with proteins are the actors, for the most part, in these deeply conserved systems. So consider then variation, an organism-specific genotype. How do we get from that genotype through to phenotype, or what you look like? Well, the variation propagates through from the DNA into the protein structure and into the system. And you can consider a phenotype as a failure of a system combined with an organismal context, an organism-specific and species-specific context that's going to get your organism-specific phenotype. And so I will illustrate a few cases of this here. All of these four morphologies are caused by the same set of underlying genes. So in humans, we have Waardenburg syndrome, which is characterized by common heterochromia of the eyes, wide set features, and commonly hearing loss. In Dalmatians, 22% of Dalmatians are deaf. It's caused by the same set of genes. Also white foal syndrome in horses, where you have an incomplete digestive tract. And also phenotypes don't have to be disease, they can be innocuous, like this particular spot on the head of certain King Charles Spaniels called the Blenheim spot was linked to the same set of genes. And so we don't have to consider all of these diseases and morphologies as completely independent separate things, as human disease and animal disease and dog disease. It's all linked to what's going on in the cell that just manifests as a macro phenotype. However, we don't just have to think about humans. So amazingly, the same set of genes also underlie this particular phenotype in plants, where it's an amazing experiment, because what you do is you take a pot of plants and you put them on their side, and a normal plant will start growing up towards the light, but a plant that has a defect in the same set of genes will just keep on growing straight. And so something deeply molecularly and conserved is going on, but we've got a different macro appearance. Our failure of systems combined with the organism context giving us our phenotypes. And so this is kind of the philosophical intro to how I'm thinking about biology. And now I'm going to go into the main work of the dissertation. So of our different conserved sequences, I'm going to be focusing on the protein complexes, the protein assemblies. So protein complexes are groups of proteins that assemble into particular three-dimensional shapes. They have a distinct geometry, and they usually form some type of functional unit, such as the proteasome, which is responsible for degrading or breaking up other proteins, or importin, which helps things get into the nucleus. These assemblies tend to be fairly deeply conserved. They're less variable than other types of protein interactions. So there's been lots of previous work in the Marcotte lab looking at discovering protein complexes and assemblies. A recent one was the panorama of ancient animal molecular complexes, where they started off looking at multiple different, to try and get an idea of what's the conserved set of complexes within animals that you expect to find in any animal cell that you look at, or any animal. And more recently, there is Kevin Drew's work integrating lots of different data in order to get a really solid estimation of what are the protein complexes of human, which is directly relevant to us. However, when I started in the lab, we, for a variety of reasons, decided to blaze off into plants. So bringing protein complex into a whole new branch of life. We realized that there was a general lack of information on certain there was a lot of information about certain plant complexes, but we felt like there was a lot that we could add with our techniques into knowing what is this set. And so we sampled from across the plant tree of life. The smallest organism is the little single-celled, let's see, oh, pointer working. Okay, the little single-celled Chlamydomonas algae. And then our more basal plants that tend to retain more ancestral characteristics, such as cilia. And then most of the plants that we're familiar with today are monocots, which is our grasses and our foods, as well as our eudicots, which is most of the rest of what we eat and see in our daily lives. So I did this in order to get a good phylogenetic spread. And incredibly, the core cell molecular functioning of the cells in these big complex organisms, you also see that already going on in this little single-celled algae. That just speaks to the conservation of these systems. So why plants specifically? I mean, Edward said I came to do synthetic biology, but what is it about this problem? So plants are foundational to our global ecological and economic systems. We need to eat in order to do pretty much anything else. However, we still don't know what most plant genes and proteins do. It's a bit of a black box, what some of these plant-specific proteins that are only found in plants that haven't been characterized in other systems or studied in plants are doing. And so there's this quote from Sue Rhee, where she says, if a stringent definition of function is used that includes biochemical activity, subcellular localization, and biological role based on experimental evidence, only about 5% of Arabidopsis genes are characterized. And this number is even lower in non-model plant species. So this is Arabidopsis. It's a commonly used model plant. There's a lot of genetic tools for working with it and like mutants available. But once you start getting into the complex crops, just the amount we know about what the proteins are doing in comparison to the amount we know about what human proteins are doing, which isn't complete either. Yeah, there's a lot left to do. So our plant complexes, they've never been systematically queried. Systematically means we want to cast a wide net and see what comes up. So we're not looking for a specific complex. We're trying to get up as much as we can at once. Also, protein interactions are a gateway to protein characterization. So say you find two proteins interacting, and one, you know what it does, and it's been studied, and you know where it is in the plant. And the other one, you have no idea, there's no data. So by virtue of finding those two in a stable complex with each other, you have a really strong prior of what the function of this second protein is. And so via protein interactions, these stable assemblies, we can propagate some of the knowledge that we already have onto all of these other new proteins. So the first step in finding protein complexes is to do biochemistry. Most of it was done and conceived by the wonderful biochemist, Dr. Ophelia Papoulis. And so we wanted to get endogenous proteins and complexes as fresh as possible with as minimal disturbance. So this took coming up with some interesting, like low soap extraction techniques for various weird species. So here you see her with some broccoli that she got at like 5 a.m. from the Essential Market grocery delivery truck. It's like a back alley broccoli deal. To the right, you see a bunch of coconuts that we had shipped from Florida, and some nice people in the lab chopped open with a machete. And it turns out coconuts are a fantastic source of high amounts of nuclei. And so just picking samples based on their phylogenetic spread, but also their utility in accessing certain cell compartments. And so in the center, you can see a lysate made from one of these species. You take the plant and you grind it up on liquid nitrogen until it's a fine powder, and then do various extractions so that the proteins of the cell, the more soluble proteins of the cell, are suspended in this solution. So then we take the solution and then flow it over some type of separating column so that over time, things drip out at the bottom of the column, and you can move this 96 well plate so that you get a sampling from this gradient over time. And then we take each of those wells and subject them to mass spectrometry, which lets you identify what proteins are in each well. So we call this process fractionation into fractions, and each of these well is a fraction because it's a fraction of the whole gradient. We tried to sample a range of tissues. We didn't want to just find leaf complexes, but we wanted to find leaf, nuclear, seed, germ, and things like that. In total, we surveyed 13 species. There were 35 separations. Each separation is one of these plates, and 2,100 fractions got total, and over 14 million peptide mass spectra, which is a large number for it's a huge number for an experiment like this. However, we've collected a bunch of these samples, and you immediately ran into a problem with protein identification. So plants tend to have more highly duplicated plant genomes compared to other organisms like humans or yeast or bacteria. Humans are diploid. We have about 20,000 genes and minimal history of whole genome duplication. However, in contrast, if we look at these common plants, each of these blue circles is an ancestral whole genome duplication. And what happens is a whole genome duplication is you have a complete doubling of genetic materials. So you could go from having 30 chromosomes to 60 chromosomes. And then it can double again and you end up with 120 chromosomes. And that's the situation you're kind of looking at in something like wheat, which is a highly duplicated sample. And so what this gets you is that instead of just having one version, like one gene, one protein, the protein does the thing, you might have 10 versions of that gene. And those 10 genes are making 10 slightly different versions of the protein. And so why is this a problem for proteomics and for telling what proteins are even in your sample? I'm going to start off with a simplified example of what you do in mass spectrometry to figure out what proteins are there. So consider these two proteins, A and B. They're fairly distinct from each other. The first step in identifying them out of a sample is to chop them up with an enzyme into smaller bits. And then you can take these smaller bits and identify the peptide sequence. And from the simplified example, you can see we've got two peptides from protein A and one from protein B, and it's clear that they're in the sample. However, consider wheat with its extensive history of genome duplication. It has 140,000 genes and is hexaploid. You might easily have a situation like this, where you've got 5 fairly similar, usually not exactly unique because you've got some mutation and variation, but many shared peptides. When you detect peptides in the mass spectrometer, which version of protein A are these peptides likely from? And so what ends up happening in practice is you get semi-random allocation of these shared peptides between different proteins, and it changes stochastically when you rerun it. And so we wanted a different solution where we could more confidently assign these shared peptides. And so we can take an advantage of a property of evolution in that these 5 protein A's ended at some point from an ancestral protein A. And so we can then consider this as a ortho group A, or orthologous proteins A within the same genome. And so now when we detect peptides, we can easily say we identify ortho group A and we don't care which particular protein it is specifically from. Good joke, Nico. So incorporating this type of evolutionary information, which we could do ahead of time and modify the mass spec lookup algorithm to respect, we drastically increase the coverage and recovery of proteins and peptides in these more highly duplicated plant species. So in wheat, in these 5 cases, we saw between 300% and 400% increase in peptide spectra, interpretable peptide spectra. And so this took the wheat samples from kind of like meh looking to gorgeous. And this type of increase in recovery held for the rest of the plant species, particularly the more highly duplicated species like soy and quinoa and maize, which had was recently a tetraploid, but it started to diploidize. The trend in whole genome duplication is you have like diploid to duplication gets you tetraploid, and then through gradual loss of genes, you kind of go back down to diploid, and then it might start again. And so it's this really interesting process. And so in the duplicated genes, you've got big recoveries, however, not really affecting the less duplicated species, like the Chlamydomonas, little algae with only 20,000 genes. And a big advantage of this approach is that we can now compare data from multiple organisms with multiple scales and repetitiveness of genomes in one coherent framework. So we pick predetermined ortho groups, here A, B, and C, and sort all of the protein sequences from all of our organisms into these bins. And so what we can then do is consider all the proteins from ortho group A, no matter what species they're from, as generally equivalent, or expecting pretty equivalent behavior, and that holds up when we check that that does happen. And so by this new orthology grouping prior to mass spectrometry approach, we get gains both in recovery, dealing with the polyploid problem, and also dealing with the data integration problem. And so I think this is a really promising new approach, and I'm going to be using it in a lot more analyses in the future. Okay, so now I'm going to give you an intro into protein complex discovery by fractionation. First, some kind of official jargon. We're deriving complexes from sets of proteins that are consistently found together following native biochemical fractionation. And so for the scientists in the crowd, you'll appreciate that this doesn't take affinity tags It doesn't take making a specific tagged genetic line, which can be take a really long time and is exceptionally hard in plants. It can take like a year or two to get a designer tagged line for wheat, and you're not going to get that for 160,000 different proteins in wheat. We don't have to use antibodies, which are fairly limited in availability for plant proteins. We can apply it to any type of sample and any type of species. So first, I'm going to start off with an analogy of a marathon. So consider this uncomfortably crowded set of marathon runners at the start of this race. Say you want to determine what groups of people are in the same party, like what groups of people are on the same like running team or came together or whatever. We can't tell that from the start when you've got this really hard, huge, complex mixture. But if you separate this crowd over time and over distance, what you're going to see is that the physically associated groups of people, aka proteins, that are together and part of the same unit are coming out of the separation at about the same time. And so that is analogous to our intact protein complex, which is the yellow, purple, blue, orange thing, traveling down the column, and it's going to drip out. As long as it stays intact, it's going to drip out at the same point. So more deep into this, we've got our proteins in our lysate of the Arabidopsis, and we're going to separate them across a column gradient, in this case from larger to smaller. And so what you can do from mass spectrometry in proteomics is we can tell what proteins are at what point in the gradient. So proteins A, B, and C are eluting together at this point in the column, and D and E are later. And so what that is it's a point of evidence towards the existence of an ABC complex or a DE complex. I'm going to demonstrate with some real data on two real protein complexes, the proteasome and the CCT chaperonin. If we look at how they separate in an Arabidopsis, which is that little green model plant, lysate, we can see that the different subunits, so 3 from each of these two complexes, they separate in the column in just about the same way, and they're related profiles, and they're different between the two different complexes. And if we do a different orthogonal separation of ion exchange, we get a different pattern, but they're still internally consistent in the two different complexes. And we can do this again from a seed lysate, different than sprout, and you get the same thing. And so this is the idea of orthogonal. Orthogonal meaning like 90 degrees different, building up this evidence. And so we don't have to do that in just Arabidopsis because we've got the nice orthogroup linking strategy. We can then concatenate on data from another species like broccoli, and then concatenating on the rest of our data from a bunch of other species, you start to build up this really distinctive profile or fingerprint of what it means to be a member of each of these complexes in our data. And so subunits from the same complex tend to have related elution behavior based on the property that they have intact traveled through a column and come out at about the same point in a gradient. So in the full data set, this is one protein, vector detection across our 32 fractionations and 12 training species. And then that extends down to 24,000 orthogroups and over 2 million protein abundance measurements. To show some complexes that we already know about and expect to see, we can see the proteasome in this broccoli ion exchange and this maize leaf. All the subunits are coherent with each other. And in a different complex, like ETF, they're coherent with each other, but have a distinct pattern. And so I'm doing more and more separations. You get your fingerprint, basically, for detecting these things. However, this is too much data to manually go through and try and kind of group things that look the same. So instead, we put it into a machine learning framework, which is going to identify sets and pairs of co-occurring proteins, pairs of proteins that are very highly related profiles. And each pair of proteins in our data set, so 24,000 by 24,000, get a co-fractionation mass spectrometry score, or a CFMS, with a score of 1, going to give you almost perfect, almost identical pair, and score of 0, meaning that there's no real correlation. We can evaluate these interactions against a set of protein complexes that we know exist or we know exist from other species, but the training didn't actually see. And we can calibrate a false discovery rate. So interactions, or 2 proteins is an interaction, with scores over 0.5 are about a 90% true positive rate for validation. And that's I think it's a pretty high score for a protein interaction screen. So now that we've got all these pairwise scores between all of these different proteins, we can start to cluster them into protein complexes. So like the proteasome has 30 components, and they're all highly related in their profiles with each other. And so I did a, no. Okay, there we go. I did a hierarchical clustering using a lock trap algorithm to get these sets of proteins. And how you read this is we've got nested circles of resolution. So 2 smaller circles define the 60S ribosome and the 40S ribosome, but the larger circle that contains both of them defines the 80S ribosome, since this is 2 individual protein complexes that come together to form a larger complex. And so it's been really nice to look at the layers of resolution. Okay, so our green circles here is a protein that was already known to be in the protein complex that I find it in our data. And so those are essentially our validations. It lines up with what was already in the literature. However, all the yellow circles are new things. And so these are proteins that have not been previously assigned to the protein interactions and complexes that we find them in. And the dark circled proteins are the proteins that are completely uncharacterized in plants, meaning there is no functional information or biochemical information on what these are doing in plants specifically. And so going around, the biologists are going to recognize a lot of these names, but this is covering a lot of the breadth of things that happen inside of the plant cell. Making proteins, replicating, making DNA, like doing photosynthesis, doing glycolysis. But the exciting part is for sure all of the yellow. This is all of the new things. All right, so all of the new things are going to be in this next part of the presentation, which is both our validation and our new discoveries. And for validation, for any technique, you want to know that it's real. When you're making predictions, you want to know that those predictions are actually useful and not just like saying some random number generator. Okay, so a first type of validation that I did was simply taking the monomeric mass or molecular weight of each of our proteins and then comparing that to the observed molecular weight that you can see by the property of the size separation columns. And so you've got your core diagonal of these proteins that are probably independent actors. They're eluting at about the same weight that you expect. But you also have this larger cloud of proteins, which are coming out of the column at a larger size than they would just individually. And this is the zone of our macromolecular complexes. Another type of validation we did was chemical cross-linking, which was, cross-linking is a chemical link between spatially close amino acids in a larger structure. and this is detectable by mass spectrometry, experiments done by Ophelia, analysis done by me. And we can look and see that co-occlusion, our coelution occurs in the same fractions that our crosslinks are being detected. So this is our peak of where we're detecting CCT chaperonin. Where we detect the intersubunit crosslinks are at the same point in the gradient, and they also are structurally realistic when you map them onto the 3D structure of the CCT chaperonin. You can see the same thing in this photosystem out of Chlamydomonas. We have a high molecular weight peak. The intersubunit crosslinks are here, and they make structural sense. And so that's a big validation that our matching profiles actually correspond to intact structure. Okay, so now getting into our this is where validation starts to blur with discovery. We have properties of protein complex. A property of protein complexes tends to be shared phenotype. So say you have protein A and protein B. Those are both independently found by different people to be involved in some phenotype. You knock them out, you get rid of them out of the plant, and the plant looks like something specific. If we find that those two proteins are in fact interacting, you can start to trace that same phenotype to the malfunction of a protein complex AB leading to that phenotype. So this both starts to provide a molecular mechanism behind some of these shared phenotypes that people have found over the years, but also allows prediction of new phenotypes, because you might find you might know what protein A does, but you have no idea what protein B does. And so you can predict when you knock out protein B that it's going to share the same phenotype as protein A. And we did some of those experiments. Really, really huge thanks to Viviana June in Jeff Chen Lab and Rachel Marcotte. Sorry, Rachel Cox. I'm looking at Edward Marcotte, Rachel Cox for helping me so much with all the manual labor that it took to grow all of these hundreds of little Arabidopsis seedlings from seed. We're just taking them out for a walk on the town here, clearly. Okay, so knockouts of genes in the same complex tend to cause the same phenotype. And what I mean by a knockout is the gene and the protein product shouldn't be functional. That's not always the case, but usually that's what we want it to be. Okay, so this is a seed pod of an Arabidopsis. This is the wild type. This is the seed pod dissected in half. And you can see all the little seeds with embryos lining it, and they're full. However, when you knock out either the gene for domino or the gene for law, you get this really distinct phenotype where many of the seeds have failed completely to develop. And so we can start to trace the mechanism of this interesting phenotype to the malfunction of this novel complex that we found we're calling domino and law. And this is also really interesting from the evolutionary standpoint in that law is a broadly conserved protein, but domino seems to be specific to plants and has a plant-specific domain fold. And so that's just, and so you can start to trace when did this protein complex come into be. That's just future questions. Okay, interactions also help us interpret already known phenotypic data. Just to introduce this particular problem of fungus blight that kills tons of crops around the world, billions of dollars. You can see this potato in this field, this half of the field is immune, and this half is sensitive. And so no one wants to grow these tomatoes. However, the mechanisms behind these types of fungal diseases are really difficult to study. You need a completely USDA approved, sealed isolation environment, because if these funguses get out into the crops, it's bad, for agriculture. And so we found two proteins which had been previously characterized to be involved in the fungal defense. Chi-B, which degrades the chitin in fungal cell walls, and OSM34, which lyses pathogen plasma membranes. And so finding this complex of the two, this is getting a bit into speculation, but we can kind of start to think of a hypothesis that this novel complex might be some type of fungus attacking machines. It punches through the cell walls and it goes through the rest of the cell membrane. And so that's the type of new avenues of research that finding these interactions really opens up. You can say, is that what's going on? Is that hypothesis right? We don't know. And finally, and most exciting, and also most stressfully personally, was the prediction of a completely new knockout phenotype. Okay, so this is a mature stem of a wild type standard Arabidopsis. It's got the main stem and it's got all of these seed pods, like we saw earlier, extending off. And one of our interactors in this new interaction, VDAC2, it's known that if you knock it out, you get a stunted plant, and also the seed pods, the siliques, fail to develop. And we found it interacting with this protein 3-beta-HSCG, and then grew up a knockout of that protein, and we find a highly related phenotype to the VDAC. And we see wild type, we've got our long extended siliques, and then we see these fertility defects in the early siliques of VDAC2, as well as our predicted phenotype in 3-beta-HSD. This was a really fun phenotype because you can see that phenotype is a spectrum. You can have the same genetics in two different, essentially clones of the same thing. And just because of stochastic noise, in some it's going to kind of go downhill, and others it's just able to recover at the end. And so this was an interesting spread in that type of phenotype. However, a really cool thing for me is we predicted a lot of our knowledge of these core cell biology things in plants. We've annotation transferred over from things that have been found in yeast or have been found in humans. But in this case, it's a prediction that might go back the other way, because these two genes, VDAC2 and 3-beta-HSD, are present in both plants and humans. And so because we find these two proteins interacting in plants, this interaction is very likely to also be occurring in human cells. And so it's a prediction kind of the other direction than we typically would predict in biology. And you can think of other interactions that kind of follow the same pattern. And it's interesting to think, what are the knockouts of these two proteins in humans going to do? It's not going to cause them to have shortened siliques. It's going to do something else. And so this is going back to our conserved systems and our organism context, giving us our phenotypes. And actually, there's no annotations for much human variation in these two proteins, and so it might be fatal from the start. And so because of that, we can study in plants to learn about human disease when it doesn't work out to create a human organism like this. Anyway, we also find large protein complexes. So I'm going to introduce the multisynthetase. Multisynthetase is an assembly of multiple tRNA synthase proteins. And it's thought to be a hub for translation because you could efficiently produce all the tRNAs you need all at once. And the tRNAs are what's going to bring over amino acids to build the protein chain. And so this is a structure of the human multisynthetase. It's actually fairly speculative. This is the only part that's been co-crystallized, but it gives you a sense of the scale and the number of proteins that go into this. So in a review of multisynthetases, they've been found to have a variety of structures across life. And in this paper review includes the quote, "A multisynthetase is not thought to exist in plants." And we excitingly are able to add in a little branch here for plants because it's definitely there. And we can start to do a comparative analysis of the structure of the core organizing parts of this kind of thematically consistent protein complex that you find across life. So the plant architecture seems to be most close with the trypanosome, which is a pathogenic small eukaryote. It makes you real sick. And we see a lot of overlap in these core organizing modules. And what we can also see some overlap with the multisynthetase of yeast and also of human. And also the cohort of tRNA synthases that are found across these different organisms, and apparently independently derived multisynthetase suggests that there is some type of functional reason for assembling this particular set in a spatially restricted manner. However, we don't know what that is, and that's another, again, avenue for research. So to start wrapping up, the major work of my PhD was this first direct large-scale determination of the protein complexes of plants that are conserved across plants. And so once again, looking at the yellow, there's tons of new avenues of research, tons of new biological systems to investigate, unexpected pairings of proteins, which when you look at the information pages for the two proteins, you wouldn't say, oh, I bet those are functionally related. And so what's really exciting about this is, I think, the unexpected pairings, where you're like, why is that? There's not enough information that currently exists to know why that is. And also the big side effect was these new frameworks for dealing with high ploidy and highly complex proteomic samples. And so we can now well detect proteins from these super duplicated samples. We could also detect proteins out of a complex multi-species sample. We just detect orthogroups and then figure out what's evidence for the proteins of the different species is there afterward. And so this orthology-based pre-grouping of proteins prior to protein inference, I think, is going to be really powerful going into the future. Finally, I made a website where our discovered protein-protein interactions and protein complexes can be queried by the community. Had a lot of nice interest, so all the data is accessible online. And so I want to thank our plant collaborators specifically, since that's most of what I've presented. Of course, Edward and Ophelia, and also Rachel Cox and Viviana June for doing all these plant experiments with me. Kevin Drew for teaching me the fundamentals of protein complex searching, and everyone else, our collaborators at Davis and Eunice. And I also wanted to throw up a picture of the lab as it was when I joined and the lab as it is now, except for a few people who have joined since the picture. And there's some people who are, stable throughout the two pictures, obviously Edward, Dan, me. But it's been amazing to see the lab grow and also maintain its personality, even though there's just different people. It's been amazing the whole time. Thank you.
Thank you very much, Claire. We'll give you applause over our few microphones here. I think we have a chance for questions from the audience, and